are you?
Not too bad. I just joined about a second ago, so.
Oh, good. Yeah, I just joined. Trying to get everything together for the meeting.
Then you can't hear my background noise?
No, it sounds perfect.
Oh, okay, because the radio's on.
Oh, I hear a little radio, yeah, but it's not distracting. I mean, if
I don't know, we listen to CBC all the time.
Okay.
They have a slight bias, but they're not too bad.
Yeah. Hi, Mayukh, and Mainak, I think, yeah.
Hi, how are you?
All right. That's good. And was that Mayukh or Mainak?
It was me, Mayukh.
Okay. Hello, Jesse.
I just put my initials for my last name next to Susan.
See,
so maybe it'll come different on the circle. I'm not sure.
It did, yeah. Susan C. Well,
it is.
Well, I don't know. You don't see it until you turn your camera off, but it probably will. I don't know. Usually that's the first initial, yeah. Yeah, it's SC, yeah.
What do you know?
Yeah.
No Y though. You only do two.
Yep. Hi, Jesse.
I don't know if you can see my curtains and my artwork. But, yeah, I'm not presenting or anything today. So,
okay.
I'm supposed to list. Oh yeah. I'm supposed to list people who can review my paper.
Which one?
The volunteers. The one for the waves.
Yeah, I mean, I could do it.
Development and waves. You could do that?
Yeah.
No, thank you. Okay. Anybody else? No?
I can offer look over like editing because I might not know the material.
Yeah, try
that. Okay.
Luigi Santella.
Try who?
Luigi Santella. I'll get her address for you.
Okay, and all right. Well, thank you. Thank you, Jesse. It's about a 3D microscope.
Hello, Tharun. So we've got, it's pretty good. I don't know if more people will be coming. I had a couple people who were interested in coming in. Welcome to the meeting. We're starting to get people for GSoC inquiring, so we're in the, I'll go through the Slack channel in a minute and kind of go through some of the new people and some of their inquiries. I think that'll be good. I also want to go over the DevoLearn issues in that. And we have some deadlines, of course, coming up. And so we have a lot of things going on today. So first thing, I guess, is the Slack channel. Let's see. I have to choose a nickname to chat here. Okay. Tharun says hi. Hi. So Tharun, yeah, he's a new person who came in due to the GSoC activities, and we have a couple other people. Let me go to the sharing my screen part here. Alright, that's actually something we'll do later. But we have, this is the OpenWorm Slack. And so we have two channels, DevoLearn and DevoWorm. And so in the DevoWorm channel, we have Akshay, who said he's interested in doing a project. I don't know, he wanted me to describe Digital Microsphere, which is the one that Susan is with one that deals with Susan's data and tiling it. And so he wanted to know a little bit about it. So we'll probably go over that. And I know that Krishna is also interested in that project. So maybe we'll talk a little bit about that in a minute. So we have, again, he wants to know a little bit about biology and a little bit about the project. So I told him if he wants to know about biology to come to the meetings. And I used to give people a reading list, but maybe that's not the best way to do it. We also have the meetings that kind of immerses people in things. So we also have Alexander Behrens and Shivank. Shivank is here. Satyam is here. So there are a couple people new people in the Slack channel. And then in the DevoLearn channel, which is different, this is more focused on the DevoLearn platform. We have Mainak and Mayukh who've been talking about this. And this is something that I want to let them talk about. This inference engine class that is going back and forth here. And then Naman is joining the conversation on that, and Krishna as well. So that's our Slack review. I just wondered if, let's see if we have in the chat. Okay, so Akshay is here. Good. Hi Akshay, how are you? I was just talking about you. So why don't we have Mainak or Mayukh, do you want to talk a little bit about the inference engine? Or the inference class?
Okay, am I audible?
Yeah.
Okay, it's not much, but like it doesn't make DevoLearn better in any way right now. But what it will do is that it's like a blueprint. Like soon we'll have much more models, maybe 7, 10, 15 models. But then we'll need a blueprint to be able to debug all the models. So it's basically a blueprint. Like until now, all the models are independent of each other. But we want them to fall under a common class. So yeah, it's just easier for the contributors that way.
Yeah, I think that would be good. I was reading it, and I was trying to figure out what you were thinking. And so this is like if we take a bunch of models and we put them into the platform, we want to be able to check to see that there's some compatibility, or that they're all basically working.
Yeah, exactly.
Yeah. All right. Yeah. And what's your timeline? Do you guys have a timeline for this, or what are people thinking?
The base class has been built, but like now we have to move the models that already exist into that base class. And soon enough, when we start adding more models, we'll write them from scratch based on that class itself, so that everything's scalable later on. Yeah.
Great. Very good. Yeah. So, any thoughts about that? Aside from Mayukh and Mainak, I guess it probably isn't much to say until it gets implemented, or like close to being implemented. But I think it's a good idea, and if you want to become, get part of the conversation, join in on the DevoLearn channel on Slack. Otherwise, we'll be looking forward to seeing some more progress on that. Let's see. Okay, maybe Akshay will want to introduce himself. So Akshay, would you like to introduce yourself to the group?
Yeah, of course. Hi all, I'm Akshay from Nair. I hail from India, in the southern part actually. So I've been contributing to open source organizations like since at least 2 years, and so I don't know if you have heard of this organization called KDE. I've been a regular contributor since like I don't know, like 1.5 years or so. And yeah, I've been mainly focusing on the website of this from the starting. Then later I moved on to this application called Kurogi, which is a drone controlling application. In that I was mainly working on GUI implementation, and also this thing, I forgot. Yeah, implementing a new protocol called Mablink for the vehicles. So yeah, that was my most of my contributions. I still contribute. I don't get time a lot for KD. I think it's time that I explore something new. So I came across INCEP and I found the project idea of digital microsphere so interesting. So yeah, that's how I came to know about the Slack channel and you guys, Bradley and Mayukh, from the mentor list. Yeah, that's how I joined the channel. Yeah, that's it.
Oh, welcome. Yeah. It looks like Ujjwal's here, correct? Yeah. Hello, Ujjwal.
Hello, hi sir.
Hi. It's been a while.
Yeah, so pretty busy with the deadlines, but I'm happy that it worked out and now I have a free slot during this time.
Okay. Yeah, I think you've been in contact with Akshay about the some of the. Well, I think he was talking maybe to someone else about the Area project.
Yeah, it's Mayukh actually.
Okay. All right. So yeah, we have actually the GSoC projects. I think the application. So they've been out in public for a while, but the application period doesn't really start until beginning of March. I think maybe March 8th or 9th. So that means that coming up, people will be making applications. They'll be writing them up. And this year, it's a little bit different. Last year, it was like a whole summer. So this year, it's like 10 weeks. So they've shortened it by about 6 weeks. And that means that it's going to be a little abbreviated in terms of how much you can get done. And then people are going to have to think about that in terms of their schedule. So, a typical GSoC application is where you have the main body of the application, and then you have, you go through, we have, who else? Shruti's here. Hello, Shruti. You have the main body of the application where you describe the problem, and then you get into you have to propose a schedule for doing it. And so this is the thing that I always go through with people every year. You walk through the schedule and you try to figure out how to implement the project. And so that's always a challenge because people want to do everything at once, or they don't know how to plan out step by step, and you have to design contingencies into the plan. So if you run against an obstacle, then you can overcome the obstacle and work on something else while you're working on the thing that you're not quite getting. And we've had this several times come up where we've had problems with getting compute power. We've had problems with some algorithm not working. So we just basically do something else for a while, and then that problem gets solved, or the person solves the problem, and then they can address that one later. So there's a lot of shifting that you have to build into the schedule. And I think that's what they're trying to go for in the program. They're trying to teach students how to do that. So we're gonna have in the coming weeks, we're gonna have an opportunity mainly in Slack, but maybe also in the meetings, to discuss this and to go through some of the mechanics of an application. And if you need me to read things over, I'll read them over. And so give you feedback on it. So I offer that to everyone who tries to apply to a project, that I'll read over their applications and give them feedback. So that's good. I'm looking forward to seeing, we have 3 projects this year. We have the Digital Microsphere, we have the Area project, and we have the DevoLearn project. So the DevoLearn project, of course, is the maintenance of DevoLearn. The project for Area is to move it to, sort of new vistas as well, improve the algorithm, improve some of the, what we're, getting out of the, what we're sort of segmenting, and we're moving more into identifying motion and things like that. And then the Digital Microsphere is this is something that Dick and Susan have worked on for a while. And this is the idea of, being able to capture an embryo from multiple perspectives and then put that together into an atlas or some sort of visualization. So this is Susan has developed this thing called a flipping microscope, which is where you take the embryo and you flip it. You're able to pull it into the water column, or into like a. You're able to pull it down in such a way so that it flips over, and you're able to like just take a camera at one perspective and image it in different, as it turns, and so you get a full rotational perspective of the embryo. And Susan, you want to talk more about the vision for this, or?
Yes, well, I have a flipping microscope, and now I also have a still ball microscope that takes images from, 90 degrees on a sphere. Then I have I have 9 microscopes. The bottom one is kind of a holder for the whatever object you're imaging. So I have, let's see if I can, so I was hoping to take pictures of living things, such as a salamander egg, such as a frog's egg, and you can look for cell behavior on the surface if you can find a microscope that will do that. And if you're looking at living cells, they work better in situ, like in their own environment. So no cutting cells off the surface and then looking at how they behave. You actually need the whole embryo or egg if you're going to look at how an embryo develops, or I don't know, bean sprouts, or just any system like that. And I hope I have a microscope that will do that now. The flipping microscope's advantage was that you were taking images of all sides of the embryo, just without any glass or anything in between. The microscope I'm developing actually has covers slide glass in between the microscope and the object, but it seems to work. So I just tried it out the other day, and it seems to work. So I've got lots of data. If someone would like to process it so that you can actually see it on a sphere, like make a digital sphere and then project these images on it, because I have tons of data. There's a limit to my time because I'm taking other courses and so on.
Hi, Susan.
Hi.
Yeah. Is the data available somewhere? Have you? Is it possible for me to have a look at it?
Yes. Where was that deposited? I gave Bradley some. Some of my data already.
I have it. I never put it into a public or a semi, like a private place. I have it on my desk, on the disk you sent me.
Oh.
But
Okay.
I can actually. I think I know where to put it now. So I can send out a link to an interested party like Akshay, and I can let him look it over. So I'll put it up this week, and I'll send it out, and we'll because I kind of figured out a solution for that. But I think that'll be good.
Well, Bradley has some of my imaging data from my flipping microscope, and as I develop the automation for my ball microscope, I'll be able to get some axolotl egg data from that too. Right now, I don't have the automation made. It's being frustrating. I'm trying to work with links to a program on Windows, and it just doesn't want to work for me the way I think it should. So,
Susan, do you have any pictures of microscopes you can show people?
Okay, let's see.
Yeah, go ahead and share
I
your
do
screen.
have I have a picture of the current one. I don't know. I need to share my screen?
Yeah.
Okay, I'll see if I can share my screen. It doesn't want to, of course.
Oh.
Like, I'm at a distance. Like, I live in the country.
Should be the lower left-hand corner of the screen.
Or you could just post the picture in the channel. That would be also helpful.
Yeah, well, okay. I'll maybe just try to upload it. Anyway, it is just a 3D printed ball with microscopes stuck in it.
Oh.
And the microscopes are like a pencil. They're kind of like this. And they're stuck in the ball at all angles. And that's basically it. It's kind of simple, but it works. At least I've got it so that it's taking pictures of a peppercorn, as Dick suggested, a peppercorn. So I'm taking pictures of a peppercorn marked up with felt markers. So yeah, I don't know. Let's see. I'll try to get an image of it up. I have one that I'm not using for publication, so I should be able to do that. But I'm not sure how to get the image up there, so. Should be able to copy and paste.
I'm not sure if you can do that. I might not be able to. You could just send them. I can send them out on. I can send them out via email if you want to do that too. It would be offline, but that would be okay. Just so that people have an idea of what it looks like.
Okay. Type. Yeah, and I can't get it to paste in the window, so.
Yeah.
It's annoying.
So what's the significance?
Oh,
yeah. What's the significance of using a peppercorn? I mean,
it's about the same size as the axolotl eggs I eventually want to image. It's about, I'm using smaller ones, so they're about 3 millimeters in diameter, and so that's the approximate field of view that I want for the eggs, because the eggs are 2 millimeters in diameter, and if I had that, then I would be able to see the egg from all sides and not have the microscope necessarily lined up perfectly, but it's hard to get them all lined up so that they're aimed at the exact same spot, so having a 3 millimeter field of view is good.
Good. Yeah, and then so the idea is to take those images of like sort of a total surface of an embryo, and then to project it onto an atlas or some sort of flat place where people can explore it. So I think Dick described it as sort of like a Google Maps or a flat map, where you take like a globe and you take the surface of the globe and you make a flat map. And you can explore a map, linearly, just by moving around the map.
Oh.
But in a sphere, of course, you'd have to rotate it and look at it. But in this case,
I like the sphere idea, because then you can rotate it and see the different parts of it, like right.
Yeah, Bradley, it's a little more complicated in that the sphere should change in time.
Okay.
It's as if you had a satellite looking at the Earth at a slow enough pace so you could watch plate tectonics.
Yeah.
Yeah, and that's why I need the automation, is so that I can get time lapse.
Yeah.
Yeah, we need the sphere versus time, in other words.
Okay, yeah. Yeah, that's good.
And we need some curious features like the ability to image what's happening around one single cell, even though the cell may be moving over the sphere.
Yeah.
Yeah, so so the goal must be like to create a GUI that supports this, or
Yeah, there would be a GUI, and of course we can do all these things, and we can figure out a way to do it in a GUI that we wouldn't be able to do, say, with a paper map or with a something that isn't a bunch of images just kind of curated. This would help. We'd basically be able. We'd have a time series of images. We'd have images from all sides of the embryo. So you'd have all those locations in space and time, and then you'd want to put them together in the GUI in such a way that people could explore them. Maybe in sort of a linear fashion, but also in a nonlinear fashion. So you want to be able to, like, if I find something at time point X and I want to see something else at time point Y, see some process going across the embryo, that people would want to be able to identify those. But to have it displayed in that way, it's I know it seems a little abstract in the call for projects. The description of the project, there are actually 2 papers that are listed there, and those are the papers that you should read or look through, and that should describe a lot of this stuff. It should make it concrete.
okay, yeah.
Okay. So that's that. I wanted to ask, does anyone want to present anything today? I don't remember who had anything. Okay. So I guess Krishna's not here today, so I'm going to show his slides that he made. He made an onboarding thing here for GSoC. And I'm going to go up to the... Here it is. So this is his onboarding slideshow that people, again, gain access to. So this is just giving people some information about open source, why you should contribute to open source developers, and kind of a cute picture about that. Then the Summer of Code kind of describes the project. So again, highlighting that it's a 10-week project this year. And then this is the schedule. So we go through from organization. Oh, that's not for us. The student application period is actually March 29th through April 13th. But the people often start earlier than that. So, but that's when you can submit them in that time window. Then the projects are announced May 17th, and then we go through community bonding and all that. So he puts the timeline in there about DevoWorm. This kind of, maybe I'll move these around because I think maybe this comes first. But I haven't had a chance to play with it. And then want to contribute to DevoLearn. So then he has a bunch of things that you can do to contribute to the project. So we have determining which part of your solution is worth contributing to, adapting the code and integrating the desired changes, extracting the relevant code fragments. These are just all different ways to contribute. Again, we might make this into like a diagram too. I have some diagrams that I've been working on in another project that I might use for this. So this is something that I don't know. We had mentioned like starting a group, an onboarding group, but I think that might be a little bit too much labor for people. So I think probably something like this would be better to do in terms of giving people, like when they come to us and ask about, how to get involved, maybe closer to the application period, we can give them this onboarding presentation. So I didn't know if Mayukh or Ujjwal had thoughts about that. Should we be doing that, or should we be doing more in terms of onboarding?
So I think that onboarding PPT is good. We can also add some slides on what are the projects in what are the different tech stacks that are being involved in the projects, so that people can get like a very clear view about the project itself.
Yeah.
Yeah, we might need to elaborate on the projects.
Yeah, I think so.
Just like the open source part is fine, but I assume that most of the people who come for GSoC, they already know about open source. So we might want to focus more on the projects themselves so that people have a better idea of what they're touching.
Yes, again, like people want to get a clear idea about project.
Yeah. Yeah. All right. Yeah, and then this is something that, we can always give them, like, a lot of information or a little bit of information. Like, the description is very short, but I think, it's... Actually, I think maybe one way to do this is to point them to the data that we have for this. So point them to resources. And maybe, like, point them... I always say point them to readings, but sometimes that's not... sometimes I just, like, throw a bunch of readings at people. It's like, what am I supposed to do with this? I guess it's just, I would... And I also probably add in something on the application process. Like what goes into a successful application.
And also like not too much like reading. I believe like just to like give a clear picture like what project is. I think it will be more than enough. Like instead of pointing data and all this, just we have to also like this is the project we are going to do, so that they can have an idea before starting. Like yes, this is the final aim of the project.
Yeah, I think that's very good. Yeah, I think that's good. So we'll update those. I'll get in touch with Krishna and update those slides, and then we'll review them maybe next week, and we'll see. We'll make sure that they're there, and this will be good for like a template for subsequent years. So we can, if we change the projects next year, we just update that stuff, and we know how to do it. So this will be good. I think this is a good way to manage when people come to the organization and ask, what are your projects and how do I get involved? To have this for people, and then we can put it in the Slack, invite people to Slack. So we've been doing that, but then when they get in the Slack, just don't dump them in the Slack. Give them some onboarding stuff. So that'll be we can do like pin that to the channel, so that's not too hard to get people involved. So yeah, I just wanted to bring that up. That was something Christian has been working on, and we, I think that'll work out. So next thing I wanted to talk about is the submissions. Actually, I have some things in the chat here. So we have Akshay. Mayukh said the ball approach is very interesting. That was referring to the digital microsphere. Dick gave this link to his paper from 2009 on the topic, and that's of course in the project description. If you have any papers related to digital microsphere, and then the other paper besides the 2009 paper is Susan's paper from 2018, in which she spells out this 4D flipping microscope. So it's where you take the microscope, it flips, you can image. The thing is, it rotates, and then you're doing this in time too. So as the embryo is sort of changing. Yeah.
Bradley, it's probably worth mentioning. The trick there is that these embryos are bottom heavy, so if you turn them upside down, they rotate automatically.
Okay. Yeah. I'll just make a note of that. And then I think that's it. There's Akshay's contact information, and then Dick invited him to some papers. So next thing I want to do is go to the submissions document here. This is, of course, the document that we're using for different things that we're doing in the group. We're submitting things to different places all the time, and we're getting this we're kind of keeping track of it here. And I saw Jesse in here last night, so he was updating things. I was updating things last night. So this again, let me put this link in the chat so people have access to it. Okay, you can follow along though here. So the first thing is here is this Evolution 2021 venue, and this is a conference. It's on evolution mainly. There's some biology, and the deadline is actually a little tricky. I thought it was March 1st originally, but I think the submissions open March 1st. This is a virtual conference, so they're trying to plan it out, I think. But the submissions open today, so I have to go to the website and check the actual submission deadline now, because I don't think they had it up until now. So this is evolutionmeetings.org. And again, we have two things we're planning for this: Kill the Winners and the Euler Paths for Life, which I've talked about in the meetings. The Kill the Winners is something that Krishna proposed in a previous meeting, and so he's working on that. This flash talk has been completed. The paper for DevoLearn is in progress. It's just kind of stuck. I haven't had a lot of time to work on it. We might think about waiting until we get some, maybe, even until we do this year's GSoC to, get it fully formed and sent out. I think it was Mainak who mentioned that his school wants him to, they have to review any publication he makes. I don't know if that was Mainak or Mayukh or...
Yeah, it was me.
Okay. So it's Mayukh. So we'll do that when the time comes. We're not quite there yet, so I'll let you know when we're close to something that we're gonna make public. So we'll be working on that. Growth form and the theory of deep learning. That's another abstract that we don't really have a home for right now, but there's going to be something that comes up that'll fit the bill for that. So we have that sort of on the back burner, but this is the document for this. So this is the shared document, and if you want to join in on that, you can contribute in some way. You can add some content or some references or do some editing, and so it's a kind of an interesting take on deep learning and how we might use deep learning to model developmental biology and vice versa. We can use developmental biology to inform deep learning. So I've noticed that there have been a couple papers that have been kind of going in this direction, so I wanted to stake out some territory here. This is the GSoC various projects. So again, we have this on, actually this time is wrong here, the deadline, but we have all the projects listed on GitHub. So that's if you're interested in, they're also on Neurostars. So I know a lot of you who have been involved in this kind of have seen the final versions of those project descriptions. Then we have bacillary and non-neuronal cognition, which is a paper that we owe. The deadline is at the end of April, so it's kind of getting pushed back in the queue. But this is the diatoms paper. This is about the cognition of diatoms, and so this is just kind of in sort of pieces right now. We have the document that we're writing up, and we have a presentation that's been made, and we have ideas, but those all have to come together in one place, and that has to be done by the end of April. So we'll be ramping that up in the next couple weeks. There's the International C. elegans Conference, which is coming up. I don't think we're going to submit anything to that. It's pretty biologically oriented, but if you want to attend this, it does cost, I think, a couple $100 maybe. Well, maybe not for students. I don't know what the registration fee is, but this is the C. elegans Conference that they have every 2 years. And so if you want to know all about C. elegans, you can attend this conference. The abstract submission and deadline is March 25th, but the registration's a bit later than that. This was something that you would have to travel to normally. So it's online now, so it's an opportunity to do this without traveling. It's usually in Los Angeles. This last year was supposed to be in Scotland, but they canceled the in-person version of it. So that's just an aside. This DevoLearn paper, actually, we have this here, so I don't know why this is a duplicate. But I'll make a note of this. This shouldn't be a duplicate, but I don't know if this is something different or what. I'll just make like a gray or something for duplicate. Then there's this Euler Paths for Life. So this is for CompleNet, which is a conference on complex networks that's coming up. This is something that has a deadline of March 26th. And this is a conference, if you're interested in complex networks, you might want to attend. There's CompleNet and then there's Networks 2021. So CompleNet is one conference, and they have, you can submit, I think, a 2 to 4 page abstract, extended abstract, and like an 8 to 12 page paper. So, being very ambitious, I'm seeing this Euler Paths for Life as a full paper, which has 8 to 12 pages in their template. And then this Embryo Networks and Connectomes, which is an abstract that I submitted to Networks 2021 already. Oops. But can also be sent to CompleNet because you can send them to multiple venues. And again, this is just to see what gets accepted and what the best venue is for it. So, as Networks 2021 is virtual, CompleNet 2021 is virtual. And if you know anyone interested in complex networks, so this is, like a lot of the complex network stuff, like brain networks and, social networks and things like that, they usually have a wide variety of things going on there, from like gene expression networks to brain networks to social networks to other really interesting types of networks that you wouldn't think exist. So it's like they're based on graph theory, and it's an intersection of big data and graph theory and complex systems. So it's always some really interesting stuff there. The Networks 2021 deadline is passed for submission, so this has been submitted. This is a paper so we have this paper on embryo networks, and we're adding into this perspective on connectomes, or C. elegans connectomes. So this, I don't know if I have the abstract listed. Oh, this is it right here. If you look at the deliverable column here after details, this is the abstract. So if you're interested in looking that over, that's where that is. So that's for that. Then we have this biosystem special issue. So we have periodicity in the embryo, and I know that Ujjwal and Jesse are both on this list of authors. So if you want to look over, I'm getting close to the resubmission point. I think we're getting there. So if you want to look it over and add things in, you're welcome to do so. It would, maybe some good editing, good eyes for editing, some, looking at what we have and thinking through things. I think that's going to help quite a bit. The boring billion. So the boring billion is actually something that we're coming up on here. So Darwin Days passed. Boring billion is the next one. So this is something that we talked about a couple weeks ago when I showed this. I showed all this stuff about plate tectonics and the evolution of life, and I showed this graphic of like a tree of life that goes way back to a couple billion years ago. And this is all based on something that Dick and myself and George Mikhailovsky are working on called the Boring Billion, and I think Ujjwal's interested in this too. This was going to be a paper in the Biosystem special issue, but it's probably now going to be a book contribution of some type. So I think Dick has more details about that.
Yeah, I wanted to ask if anybody listening has been trained in bioinformatics, because we may be doing some of that analysis.
Yeah, I'm not sure. I think Ujjwal maybe and Krishna have done some bioinformatics. I know because they showed me some stuff, but like this is sequence analysis, so to be clear what bioinformatics means. Because we have the data about the tree of life, and we have some information from the literature, but it's very, sometimes it's very sketchy as to what is actually going on this time. Like, people have different time estimates for things, and so it would help if we could get some sequence data and analyze it. And I've done some bioinformatics, but it might help if someone else someone wanted to get involved in that part of it. And we could do an analysis of data. There's a lot of open data out there, secondary sequence data, that can be used for this. It's not very hard to get. So this is Jesse. I will be going more over the periodicity paper later this week. I am also interested in the Boring Billion. Is that still open? Yes, it's open to contributions. So we should maybe get in touch about that on Slack or email.
Yeah, Bradley, it might be a good idea to define what the Boring Billion means.
Yeah. So, all right, yeah, so people, most of you probably don't know what it is. So the Boring Billion is this period where you have sort of the beginnings of life, and then for like a billion years, nothing much happens. Like, it's pretty simple. Life is pretty simple. It's maybe unicellular, and you're just hanging out. There are a lot of theories about the origins of life, but, for like over a billion years, it just remains very simple. And then all of a sudden you get this explosion of diversity where you get multicellularity and you get different kingdoms, like you get animals and plants and protists and other types of organisms that have different cell types. And so it just happens all at once. And there's some theories about like the Earth's atmosphere, like there are oxygenation events that sort of set up sort of the end of the Boring Billion. So you have like limited oxygen on the planet for a long time, and then all of a sudden at the end of this period you seem to get this oxygenation event that really you get a lot of oxygen relative to what was there before in the atmosphere, so that allows life to diversify. And so that's what we mean by Boring Billion. It's boring because there's not, it's just life basically looks all the same, or maybe there's like maybe two species on the planet and doing boring things. They're not really behaving in any meaningful way. I mean, they're moving around maybe, but it's very very simple life. And then after that you get this explosion of different kingdoms of life and different ways of surviving. So that's what Boring Billion means. And we've worked out a lot of the sort of we haven't worked it out on our own. We've kind of like assembled a lot of information about what's going on at that time. So the contributions, I guess we have a manuscript that's being worked on. We have some graphics, and we have maybe some data analysis that needs to be done, especially on the sequence data. So if you're interested in the sequence data analysis, let us know, and we can get onto that. Then, and the final destination for this is now to be announced. It's probably going to be, we're working on a book proposal on this topic a long time ago, but I don't know, there are always opportunities to put it somewhere, so it's not like we won't be able to publish it. Then there's this Kindle book, which is something that Krishna proposed, and this is something where we take a lot of the materials that we have on machine learning and biology and put them into a book. We're still working on the details on that. Okay, there's this INCF Neuroinformatics Assembly. I don't know if I mentioned this, but INCF has something called a Neuroinformatics Assembly. It's the annual conference. So INCF, aside from hosting GSoC, they also do a lot of stuff now with sort of neuroinformatics support. So there are a lot of groups that are putting together different toolkits and different community standards and things like that for neuroinformatics. So, there's that opportunity to interact with those groups of people. And they've actually, if you go to their website, you'll see that they're doing a lot more than just GSoC sponsorship. They're doing a lot of this sort of informatics support. And they have a conference coming up. The deadline for submitting an abstract is March 31st. And this is the abstract submission portal. It needs to be 1,500 characters, which is small. It's like maybe 200 to 300 words at most. And I'm thinking of some sort of DevoZoo epistemological directories mashup. And the epistemological directories, Jesse knows about this. This is something I'm working on in my other group. It's an educational tool where, it provides tools for people to sort of do self-guided education and to get up to speed in different fields. So we can maybe present on something along these lines to this group, and maybe we'll get some good feedback on that. Finally, there's this ANN's BNN's extended abstract. This is something that Krishna proposed for ALIFE 2021. This is ALIFE is coming up. It's a conference on artificial life, so it's computational approaches to evolution and development. And this extended abstract would be about 2 to 4 pages in their template, and it's due pretty soon, so we're working on that. If people are interested in looking it over or being involved, let us know, but this is a pretty short turnaround, so I don't know what the actual opportunities are on this. But anyways, if you have anything to add to this list, let me know. Put them on the list, or send me something and I'll put it on the list, and we'll review it next week. And the reason I do this every week, of course, is because the deadlines are always kind of marching towards us. So that's good. Let's see what else is in the chat here. Okay, so this is the... Okay, so Dick says, the Boring Billion book could be part of Astrobiology Perspectives on Life of the Universe book series. So this is a book series that's coming out. And Akshay said that his invite is not working. And Jesse's email. So Jesse, put your email in there. Alright, so that's good. What else do we need to do here? Okay, this is actually the ANN's BNN's abstract. And I was kind of hoping Krishna would come to the meeting. And actually Jesse is on this as well. So this is this was a presentation that Krishna and I gave to Neuromatch conference. So this is about how taking artificial neural networks, comparing them to biological neural networks, and looking at some key differences. I'm not sure. We may have to refocus this a bit towards some specific argument, but I think it's in the right direction. I don't know if this is something that'll fit into the conference very well or not, but we can't just turn this into a catalog of things. We need to kind of get into some sort of argument, and maybe, the ALIFE conference is very interested in sort of the emergence of biological systems, so we might focus on that a bit. So this will be, hopefully by this time next week, the deadline will have passed and we'll have a completed extended abstract to show. So this is the evolution presentation, so I don't have that much to say on that, but Krishna has been working on it. So finally we have our paper queue here. I was going to talk a little bit about some of the I dug up some things on some cellular automata that I had been working on a long time ago, but I'm not going to talk about that today. I think I mentioned Shruti that she was asking about different models that we might use for DevoLearn other than machine learning, or different types of models we might use. And I told her we have a couple of models in place right now, but we can always add machine learning models onto what we have. But there are also other opportunities to do other types of models. Our group has a long-term interest, I think, in cellular automata. We also are interested in other types of models, like more general agent-based models, things like that. So these are things that you can, if you're interested in contributing, we can find a way to, put it into this model zoo. And we, it's not the types of agent-based models and cellular automata that we use are not really meant to do like data analysis. They're more meant for simulating processes. So, simulating like morphogenesis with a cellular automata. So these are things that, we can talk about later. I was going to present on it a little bit, but I don't think I'm going to do that today. Okay. So yeah, that's okay. So let me go back to the papers. All right, here we go. So I had a couple papers I wanted to review. Actually, here's a historical point here. Ernst Haeckel, who was a 19th century embryologist. I think we've talked about 19th century embryology in the group before. So February 16th, 1834. This is obviously something I bookmarked 2 weeks ago, but I'm just getting to it now. Birthday of German naturalist and artist Ernst Haeckel. So this is Ernst Haeckel, and he did a lot of stuff with embryos and kind of coming up with ideas about embryology, some of which were wrong, but some of which were pretty spot on. He came up with, capitulation of phylogeny, the idea that, embryos sort of development from sort of a very simple embryo to, to an adult actually mimics evolution. And so he came up with the concept of neoteny, if you've heard of that. And so these are ideas that, you come up with, and they, over time, they get proven to be true or false. They get falsified or they get confirmed. An early supporter of Darwin, he studied microfossils and deep-sea sediments, believing in the unity of science and art. There is even a reference in the anime Ghost in the Shell, which is this here. Anime, of course, is the animation style. So I don't know what that reference is, but that was what they were highlighting. And then this is a thing on Ernst Haeckel and the Spirit of Matter, which is something that's translated from German, so it's a bit like rough. The translation is a bit rough, but nature always provides new forms that gives new material for reflection, drawing, and writing. And they talk about young doctor with a microscope. He brought a novel microscope with a water immersion lens that could be used to look at living microorganisms. And young scholar wanted to spend half a year studying jellyfish in Florence, also mollusks and plankton. Decades later, a work of the century was to be created in the end. The young physician and natural observer was Ernst Haeckel, who was born February 16, 1834 in Potsdam, Germany. At the request of his father, he studied medicine, but on excursions with the anatomist Johannes Müller, he discovered the fascinating world of marine animals. So he's particularly fond of mollusks and radiolaria, which are microorganisms from the ocean, or from the coastal regions too. In the course of his career, Haeckel collected, studied, and drew well over 4,000 types of radiolaria. The filigree mostly radiosymmetrical skeletons of silica and dioxide. Silica had only been discovered a few years earlier. So he did a lot of these sketches. And so there's one thing about, Haeckel's legacy are these amazing drawings that he did. And of course, in the 19th century, you couldn't take a lot of high-resolution photographs, so you did a lot of drawings. And so these are some examples here of different drawings. And so they did a lot of field work as well. And this is a world distribution map. So, they would make these maps of how different species and marine sediments were distributed around the world. Gave people a good idea of what biodiversity looked like around the world. And of course, this is something that's constantly being updated as we get more data. So this is a nice article. Again, this is sort of Haeckel's pedigree. Again, this is his idea of evolution, which is not what we think of today. This is sort of like where humans are at the pinnacle, and there's this straight tree that goes up from a base here. So a lot of these ideas were sort of formative, but they weren't correct in the way we understand them today. And finally, here's this reference to the anime character. I don't know much about this, but it's, you don't get too many references to scientific things in comics. But when you do, it's interesting. It's an aside. So I'll talk about a couple papers. If you have to go at the top of the hour here, it's fine. But I will mention this here. This is Milton Tan, who's at the University of Illinois. He's an ichthyologist. He had a questionnaire. A student in a discussion asked a philosophical question. If systematics, which is the study of these evolutionary trees and the classification of organisms, if systematics is partially about classification and identifying population structure is also kind of a classification, what is systematics? Where is the line? And so that's an interesting point, because we think about evolution as, maybe like classifying traits or classifying organisms, but we also have things like population structure. So like traits exist in different places in the species range or things like that. And there are all sorts of things you can think of as like that are classification that aren't necessarily directly tied to the analysis of evolution. So, there's a lot of interesting areas there. But maybe that's over some people's heads, but it's an interesting question we might follow up on. So the first paper I wanted to talk about it's a new article from James Briscoe's group. I don't know, many of these people aside from who James Briscoe is, but this is precision of tissue patterning is controlled by dynamical properties of gene regulatory networks. And this is brand new. We've talked about gene regulatory networks in the group, and I really need to give a talk on this because I think it's important to know what exactly is going on with these things. They're kind of mysterious, but they're actually good models for looking at, like, how development proceeds, and they can be modeled computationally. But that's for another time. The abstract reads, during development, gene regulatory networks allocate cell fates by partitioning tissues into spatially organized domains of gene expression. How the sharp boundaries that delineate these expression patterns arise, despite the stochasticity associated with gene regulation, is poorly understood. So you have these, when genes are expressed, they're expressed using these networks, and you have basically a promoter which turns the gene on and off, and also regulates how much of the gene's product is produced. These products then are expressed locally in the environment and within the cell, and they kind of determine where this, they kind of are expressed spatially, so they're expressed maybe in stripes or in different patches, and they are local, so there's a local signal, and you get these, that's how you end up with patterning. You get these, the expression of genes that are specific to certain gene products that then are spatially segregated, and you end up with these patterns. Gene expression is not deterministic. There's a stochastic component, meaning that it turns on and off sometimes quite unpredictably. But despite that, we can still have these patterns emerge that look highly ordered. That's what they're trying to say. And you have these very precise boundaries. And there are reasons for that that maybe we can talk about in another week. But basically you have, boundaries that are pretty precise. You would think that if it's just this general process that it would be less precise than it is. So this is achieved not by reducing noise in individual genes, but by the configuration of the network modulating the ability of stochastic fluctuations to initiate gene expression changes. We use a computational screen to identify network properties that influence boundary precision, revealing two dynamical mechanisms by which small gene circuits attenuate the effect of noise in order to increase patterning precision. The results highlight design principles for gene regulatory networks that produce these precise patterns. So the paper actually is about looking at these networks using a computational screen, which is using like bioinformatics to look at different modes of gene expression, and then kind of figuring out how these models or how these circuits work. So they actually use an SDE model, which is a stochastic differential equation, to simulate a PAX6 mutant, which is where they knock out a gene in this mutant. And PAX6, I think, is involved in eye formation. But so they're able to use these. They want to simulate mutant genotypes and their effect on on gene expression. So a stochastic differential equation is a differential equation where you have some stochastic element in it, meaning that you have some factor in the equation that allows you to simulate things not in a deterministic way, but introduces noise in it, systematic noise. And so they're able to use these in these gene regulatory networks, and they're able to actually model this. I don't know if they have any good figures in this paper, but I don't know where the figures would be. Oh, at the end, okay. So this is, see if they have any good pictures of networks. This paper is mostly the biology part. This shows kind of the boundaries of gene expression. So you have these different stripes. So these cells are expressing this gene here, PAX6, and then there's OLEG2 down here, and then NKX2 down at the bottom. And you can see that these boundaries are pretty sharp. And there are these theories about gradients where you have different gradients of gene expression of these gene products that collide, and then where they kind of join together, they kind of form this sharp gradient, this sharp boundary. So it's not not a smooth gradation, and you get these stripes as a result. Let's see, figure 2, you have, this is figure 4. So they kind of talk about the configuration of gene expression fluctuations. So they're looking at these dynamical systems graphs, where they're looking at transitions between different states in the wild type and then the mutant. And then this is a sort of a landscape model where they're looking at these different states of gene expression. So they do a lot of modeling. It's very modeling heavy, this paper. And then they can actually, at the end of this paper, they have a glossary of dynamical systems terminology, because this was published in development, so it's going to be very difficult for some people to follow if you don't have a background in it. So if you're interested in dynamical systems, there's a nice glossary at the end here. So you have phase space, stochastic systems, deterministic systems, and if you're interested in like machine learning, you may have heard the term stochastic before, but that's actually not quite the way they're using it here. They're talking about it in a very specific way to systems biology. So a lot of this, a lot of the stochastic components of in like gene expression involve fluctuations of concentration. So if you have concentration of molecules, those concentrations fluctuate. That's sort of the essence of a stochastic approach. You also have noise-driven transitions. So you have fluctuations in concentration near stable fixed points, and these fluctuations can push the system to a different state just by, through a lot of the, through not just being not being deterministic. So, you think about like if you're swerving on the road, you might go off the road just by accident because the amount of fluctuation is, either very tightly organized around your local state or you drift into another state. And so there are a lot of things there are a lot of concepts like that that, if you're interested in reading about that, this is a nice way to do that. Another paper is this Mechanotransduction tuning stem cell fate. And so this is something we don't usually talk about stem cells a lot in this group, but so the abstract here is, it is a general concern that the success of regenerative medicine-based applications is based on the ability to recapitulate the molecular events that allow stem cells to repair the damaged tissue/organ. So to this end, biomaterials are designed to display properties that, in a precise and physiologic-like fashion, could drive stem cell fate both in vitro and in vivo. The rationale is that stem cells are highly sensitive to forces and that they may convert mechanical stimuli into a chemical response. So I think we've talked about this where you're doing things to cells, mechanically constraining them, and other things, putting them on different substrates. And that actually has an effect on the chemical response, on the gene expression response. So these cells, when they experience different physical like perturbations or physical forces that, people have done experiments where they've looked at applying different physical forces to these cells, and they're able to get them to express genes that, maybe make them jump to a different state, a different cell state. So that's basically the idea. So this is a review article that describes the novelties on stem cells and biomaterial interactions, with a more focused implication of the mechanical stimulation and mechanotransduction. So what they're interested in here is this process of mechanotransduction. So you have mechanical forces acting on the cells, and it's being transduced into this, to the cells or the physiological response, which is to express certain genes in response to those forces. And so it sounds actually like perception. It sounds like something like human perception, where we take in information and we produce behavior. And that's sort of what it is, but, this is where, people start talking about cellular cognition. There have been papers written on that topic where they talk about how cells are making decisions, cells are like perceiving things in their environment. And this is sort of what they're talking about. So this paper has this interest in regenerative medicine, which it deals with tissues and organ replacement, tissue engineering, and stem cells, which are cells that they have the stem fate, but they can differentiate into other different types of cells. And so they're also self-renewing, so they don't go through things like, they don't go through cell death. So they're able to replicate as many times as they do. There is no limit on that. In some somatic cells, like in your skin cells or your heart cells or your muscle cells, they live for a finite number of divisions before they die. So these are things that people have been interested in stem cells, and we can look at stem cells as models for this because stem cells are particularly susceptible to these signals. So they go, and then they talk about embryonic stem cells, adult stem cells, induced pluripotent stem cells, which are stem cells that you can create by putting genes into the cell and forcing expression of those genes, and it actually triggers the gene expression that mimics some sort of differentiated cell or some sort of stem cell. So you can take a skin cell, for example, put in factors that allow for, the expression of genes that then trigger a cascade of things underneath it in terms of the gene expression network, and it produces this stem cell. It becomes a stem cell, basically. it sounds magical, and it's not something that's an exact science, but people have done a lot of experiments in this area. If you're interested in this topic, this is a good review, I think, to read. And this kind of shows you an example of mechanotransduction. So you have these things going on in the environment: compression, fluid shear stress, gravity, tension, hydrostatic pressure, which is a water-based pressure, and you have all these things acting upon the cell. And so you have the cell membrane here, which has receptors, integrins, has what they call a cytoskeleton, and all these things that are on the outside. And these forces act upon this cytoskeleton and these receptors in a way that triggers different things downstream here. So there are all these things that can be triggered by forces or by things going on in these through these receptors or these channels. Different fluxes of ions, different types of other things going on, stress responses, and then that goes down and activates transcription factors, which are the expression of genes. So they basically tell the promoter of a gene to express, because the conditions are such that they need to express. And these are things that happen normally during times of stress, or maybe during times of development when they need to be expressed, but you can actually mimic these experimentally. And so you have all these things happening in the cytoplasm, they trigger things happening in the nucleus, and then these gene products are expressed in the nucleus, and then they change things going on in the cell. So that's basically the idea here. So I think that's it for this paper. And then finally, I kind of wanted to get to this paper. This is kind of a hardcore bioinformatics paper, data-driven, but I think someone sent this to me to review. Data-driven biological network alignment that uses topological sequence and functional information. So this is a paper. So the abstract here is they're talking about something called network alignment, which is network alignment can transfer functional knowledge between species conserved biological network regions. Traditional NA assumes that it is topological similarity, which is a matching between network, the shape of the networks, between regions that corresponds to a region's functional relatedness. However, we recently found that functionally unrelated proteins are as topologically similar as unrelated proteins are as topologically similar as functionally related proteins. So we redefined NA as a data-driven method called TARA, which learns from the network and protein functional data and tries to find what kind of topological relatedness rather than similarity between proteins corresponds to their functional relatedness. So what they're doing is they're looking at the network's topology, and they're looking at the relatedness of two different topologies. So it's kind of like the relatedness of two different species or two different species trees. It's a similarity, but it's really not. They're using different types of criterion for that to determine relatedness. Between proteins corresponds to their functional relatedness. So you're looking at two networks. You're looking at a functional relatedness, and then you're looking at this topological relatedness. And the topology is based on different aspects of the proteins. So this is, these are two different aspects of protein action. And so this is like, one thing is how you represent it in a graph. The other is the actual functional relatedness of the proteins. And this functional relatedness of the proteins isn't represented in a graph. So you're picking up things in the graph representation that are a bit different than the things you kind of know about the proteins functionally. So Tara used topological information within each network, but not sequence information between proteins across networks. Yet Tara yielded higher protein functional prediction accuracy than existing NA methods, even those that use both topological and sequence information. And so this is something, basically what they're doing here, is they're looking at combining different techniques and looking at how, those different techniques can be combined to get a better result. And so, conclusions, as such, combining research knowledge from different domains is promising. Overall, improvements in protein functional prediction have biomedical implications. For example, allowing researchers to better understand how cancer progresses or how humans age. And so this is, that's really the reason why they're doing a lot of this sort of stuff. So if we look through the paper, we can see that they kind of describe their sort of what they're doing here. They review these NA methods, and then they propose this better method. They show what these protein-protein interaction networks look like. So these aren't based on function. These are just based on maybe like proximity or, coincidental interactions that they pick up. So these are not exactly the same as function. So you're actually looking at these interaction networks, which tell you a lot about the global state of proteins in a cell. Like, if you want to know like what proteins are associated with one another, if you look at a sample and you say these proteins are in the same place, they're doing something we don't know what. Then you can say, okay, this is what our network looks like. But it doesn't tell you anything specifically about function. You actually have to go and do the functional assay separately. But then that doesn't fit into this global view of what you're doing. So this is what they're trying to do, is to combine these two approaches, this sort of holistic network approach, and then taking what we know about the functional relationships between different proteins and putting them together in a single model. So this is classic bioinformatics stuff. They have a pipeline where they actually look at like different protein-protein interaction networks. They do feature extraction, they do classification, they do alignments, and then they come up with this prediction. That's basically the way they do most bioinformatics. They use a lot of Go terms to look at like function, which Go terms are descriptions of function that people have found in the literature, and they have a sort of a work, they call it an ontology, which is something they work out to sort of give a very strong predictive description of the cell, what kinds of things are happening in the cell or in the protein. And so I'm not going to go any more into the paper, but I think if you're interested in it, you can read more. I just wanted to give you an example of what a typical bioinformatics study looks like. So let's see, we have a lot of things going on in the chat here. So we have Akshay says, I have to leave now. We'll meet you all again next week. Well, thank you, Akshay, for attending the meeting. Hopefully some progress on my side. So next week, Akshay and Ujjwal, I think, are interested in presenting on some work they've been doing on the Basilaria. So I look forward to that. Let's see, we have another couple of comments here if I can get back up to the top. Okay, Susan says, the gene networks probably have something to do with the mechanics as in pressure and tension. So I think that was from the first paper. Then Dick says, differentiation waves may control these networks. And then Susan says, waves are good at producing tension and compression. Dick says, I have suggested we list all papers discussed here and cited an EndNote Basic, which is free. So let's keep up on the EndNote. I know Jesse is interested in the Zotero for the other group, but I told him that we were doing an EndNote library for these papers. So I need to go back and pull out those references too. Dick also says stem cell relationship to differentiation waves, and he has this paper here on the diseased breast lobe in the context of X chromosome inactivation and differentiation waves. And then this other paper on reverse engineering the mechanical and molecular pathways in stem cell morphogenesis, and a cell state splitter and differentiation wave working model for embryonic stem cell development. So those are all, these last two I was thinking about when I was reviewing the paper on the different forces, the mechanotransduction, and then the stem cell papers. So that's a very good set of papers to read if you're interested in what some of those things might be doing in the cell, and especially in terms of differentiation. Mainak says, interesting paper. Thank you, Mainak. So Ujjwal says, Sir, I and Tharun are working on Basilaria. I haven't talked with Akshay yet. Oh, okay. So, sorry, Tharun was the person who was working on the Basilaria, and Akshay was working on I'm sorry, I forgot. But anyways, I look forward to the presentation next week on that. I know that you've been previewing some of that in the Slack, so keep up the good work. And again, if anyone's interested in, if anyone has a paper they want me to present, or they want to present, we can do that. And just send it to me, or before the meeting we can pull it up. Or if you're interested in presenting on some work that you're doing, let me know, and I can put you on the schedule. So, does anyone else have any questions before we go? Or comments? Ah, Markus. Markus is in the meeting here.
I'll send you a picture of the microscope.
Yes, that would be good. Thank you for that. And I can make that, we'll put that together more coherent, just like something that would help people really capture their imagination. I think that'd be good. So, Markus, did you want to, I know this is kind of the end of the meeting, but did you want to introduce yourself? Oh, hello, yes. Oh, go ahead. So Markus is a graduate student from Germany and probably won't be able to contribute too much as of now. Well, that's fine. Everyone here is, some people will contribute a lot, some people are just watching the show, but it's fine. So I'm interested in the topic, and Nick asks what field. Embedded systems. So is this like biological systems or systems in general, or? IT, okay. I mean, we do a lot of machine learning, computational stuff. We do a lot of biology, but we also do a lot of, in crossing those lines, there are a lot of things things like embedded systems that we deal with. So yeah, we might, why don't you, come to our next meeting, and we have a YouTube channel, which is also, it's the DevoWorm YouTube channel, so, which gives you a lot of information about past meetings. So if you're interested, browse that and see if maybe there's something you'd be interested in doing, or just seeing what we've been talking about. Okay, that sounds good. Well, thank you for attending, everyone. I look forward to seeing you next week. And if you need to contact, please use the Slack and the email channels to discuss some of these things further. Okay, have a good week.
You too, bye.
All right, bye.